the projects I pick always turn out to have medical importance. The projects I pick to study. That's my background. And I started out by trying to understand how penicillin kills bacteria. And if you pick up textbooks, there's my first 15 years of work on how penicillin kills bacteria. So I did that work at a lot of places, NIH, Wisconsin, Washington University in St. Louis. And when I moved to Harvard in 1967, I decided that the penicillin problem was almost finished, and I needed to find something that would keep me excited for another 20 or 30 years. So I decided to study transplantation antigens. And it was a time when kidney grafts had been established and heart grafts were just first being done. And no one understood why grafts get rejected. So that was the problem I decided to tackle, why grafts rejected. And I worked with a colleague. First I worked by myself for 10 or 15 years, and then with a colleague named Don Wiley, who you probably saw in the newspapers 2.5 years ago. He died accidentally at a very young age in a fall from a bridge in Memphis, Tennessee. It's on my mind because I'm going to the International Congress of Immunology tomorrow. And there's a memorial lecture, and I have to do the introduction to and one of our students is giving a lecture. She's now, anyway, so together we solved the problem of what transplantation rejection is. It has to do with proteins called histocompatibility proteins. Do you know about them? Raise your hand if you ever heard of histocompatibility. Oh, great. So we figured out what they're for, what they look like, and why grafts are rejected. What they're for is to protect the population from catastrophic variations in infectious agents. They present peptides derived from foreign proteins to the immune system. And I don't think I have a slide of a histocompatibility protein.
The slides are ready, by the way. They're on them. They're ready.
No, I don't think I have one in this set. So it's an immune rejection phenomenon, and then I got interested in immunology. And that's what I'm supposed to talk about today. Is that right?
Yeah.
Yeah.
Natural NK cells?
NK cells are part of the immune system. Has anyone talked to you about immunity?
Yeah.
Who's talked to you about immunity?
Arn.
Who?
Arn, Sparnbach, Glimker.
Laurie Glimcher?
Yeah.
Oh, did she come over here and talk?
Yep.
Yeah.
And who was the first one?
Arn.
Spell the name.
H-A-R.
No, I don't know him. What did Laurie talk about?
T helper, differentiation.
Yeah.
TH1, TH2.
TH1, TH2 cells. Okay. Does anyone talk to you about antigen presentation? So that's the side of it that Don Wiley and I worked on, how foreign substances get presented to the immune system. I'm leading up to natural killer cells. So how foreign substances, they get foreign, let's say you get infected with flu virus. How does your body recognize that you're infected and fight it off? And so the proteins of the virus are mostly used to assemble virus particles. Virus has nucleic acid in it. The nucleic acid I think it's RNA in the case of flu virus, but it gets made into DNA, and then it makes proteins, and the proteins are mainly used to make new virus, but some of the proteins get chopped up, and they get presented to the immune system by these molecules that Don Wiley and I studied called histocompatibility proteins. So, do I have a picture of one? I don't think I have a picture of one. Have you ever seen a histocompatibility protein?
Just diagrams.
Have you seen diagrams? That's all I've ever seen. And crystals. I can see the crystals. Have you seen diagrams? Have you seen a peptide in the class? Oh, you've seen that stuff. Okay, that's the stuff that Don Wiley and I did. So they're very polymorphic. What's that mean?
Many shapes.
What?
Many forms.
How many do you think there are?
Lots.
What's a lot?
Hundreds of millions.
What?
Hundreds of millions.
Not hundreds of millions. Well, there's enough so that, except for identical twins, there are probably no two people on the Earth who have exactly the same histocompatibility protein. And that's why grafts get rejected. Because they're recognized as foreign. If you have the graft has a new histocompatibility protein, it gets recognized as foreign and gets zapped. The cells get zapped by T cells that Laurie talked to you about. So the polymorphism means that these proteins bind in the population an infinite variety of peptides. There are actually 6 loci. You know what a locus is? Locus is a place where a gene is encoded. There are 6 loci, so you have 6 histocompatibility proteins encoded on each chromosome. So that means you actually have 12 different on your cells. So when a virus starts mutating, there's some people who can recognize the mutant peptide. So that means no variation in an infectious agent is going to wipe out the whole population on Earth. So this system didn't vary to protect individuals, it varied to protect populations. So if you think about the flu pandemic in early part of the century, last century, around 1918, 1920, I forget what the mortality was, but it was very high. But there were some people who survived. And we presume that they had different histocompatibility proteins that allowed them to provide protection against that variant of the flu virus. So, okay, so you have 6 loci, just to show you how varied the system is. There are from 100 to 400 alleles, varieties at each locus in the world population. Well, one of the problems is that the world population essentially means the white Caucasian population, because that's the population that's been mainly studied. So just now we're beginning, I'm not in that field anymore, people are beginning to look at New Guinea natives, Arabic populations. And so the number 100 to 400 alleles at each locus is still counting. There's 10 added around every month. There's a committee that accepts that this is a new allele, and they publish a little newsletter. So my memory is there's around 10 every month that are being added to the list. So one at each locus if there are 12 loci. Let's get back to blood cells. So the B cells in the blood. Has anyone told you about dendritic cells?
Yes. Yeah.
We've talked about that.
Most of our speakers,
Not everybody.
most of the speakers have mentioned them.
Yeah, they're the main antigen-presenting cells, but they represent only about 1%, maybe less, of total white cells in the organism. Has anyone shown you a picture of dendritic cells in the skin? Langerhans cells, have you seen that? They're so beautiful. They're arranged in a pattern. They're arranged in a pattern. Your whole skin is protected by dendritic cells called Langerhans cells. He was an Austrian pathologist, I think, who first described them. They're in a regular pattern. When you get an infection in your skin, the Langerhans cell picks up the foreign substance, and picking it up activates the Langerhans cells, then moves to a lymph node, and then activates T cells, and you get immune to that infectious agent. So blood, spleen, lymph nodes are full of a large variety of different kinds of cells. Some of them, like dendritic cells and B cells, can present foreign antigens to the immune system. Other cells, like T cells and natural killer cells, are the effectors. They do the work. They recognize the foreign antigen and decide what to do about it. Then still other cells, polymorphonuclear leukocytes and macrophages, they're part of the innate immune system. They don't need to be activated. They're already active, and they take up invading bacteria. Okay, so natural killer cells, what are they for? They were ignored for a long time while all this work was on histocompatibility proteins, on antigen presentation, on T cells was going on. They were discovered in Sweden and at NIH, mainly in Sweden, as cells in the blood that would kill tumors. So if you take a mouse tumor and incubate it with natural killer cells, it's likely not to be killed. But if you if you inject a normal tumor, like called RMA, is the cell line, the experiments were done with, under the skin of a mouse, the tumor will grow, and nothing will kill the mouse. But if you make a mutant of RMA, which is called RMAS, then the tumor won't grow. The natural killer cells will attack it. So RMAS is a mutant that doesn't have any histocompatibility proteins on the surface. So natural killer cells were first discovered as cells that are able to kill tumor cells that lack histocompatibility proteins. And they're probably very important in immune surveillance of tumors, although I find that literature somewhat wishy-washy. So let's take an example of a melanoma, a skin tumor arising from a mole. That tumor will metastasize and grow in the liver, and that's why in the end you get killed. If you look at the metastatic lesion, the tumor itself may have cells with histocompatibility proteins on them. But the cells in the metastatic lesion are devoid, often devoid of histocompatibility proteins. So you could say that the original lesion is held in check by the immune system, which recognizes something, some protein from the melanoma that's presented by the histocompatibility proteins. But the cells in the lesion don't have histocompatibility proteins. And they're targets for natural killer cells. So natural killer cells can keep those metastatic lesions in check for years. Do you know the natural history of melanoma? Have you ever known them? There will be seeds in various organs, and you can't find them. And those seeds are kept in check by the immune system for as long as 15 years. And then suddenly something happens and they begin to grow. And we don't really know what happens. But one possibility is that they've acquired another mutation that makes them no longer susceptible to natural killer cells. Something happens to the membrane. Something else happens that prevents the immune system from controlling those foreign cells. So that's one function of natural killer cells. Let's see the first slide. Yeah, so there are the functions. Immunosurveillance of cells that have downregulated expression of class 1 proteins. There are two kinds of cells in which class 1 proteins are downregulated. One of them is virus-infected cells. So if you have that slide in here.
They're all.
I may have a slide, but in the first week after a virus infection, the virus titers held in check by the natural immune system, while the adaptive immune system is revving up. So T cells take a week to expand clonally, and then they can eliminate the infection. But if there wasn't something to keep the infection in check during that first week, you'd be dead before the adaptive immune system could take over the protective function. So natural killer cells function in eliminating virus-infected cells during the first week of infection. Now, viruses want to escape the adaptive immune system. And so they've developed mechanisms to get rid of histocompatibility proteins on the surface. There are, of cells that they've infected, there are many mechanisms that are involved in getting rid of histocompatibility proteins from the surface by viruses. HIV does it, for example. But HIV does something very clever. Maybe there's slides. It does something very clever. There are 3 histocompatibility proteins, class 1, called A, B, and C, and HLA A, B, and C. And C has a special function that I'm going to tell you about probably on the next slide. The function of C is to inhibit natural killer cells. A and B present peptides to the immune system, but C's function is different. Its function is to inhibit the activity of natural killer cells. So if you were HIV and you wanted to escape from the immune system, what would you do?
Destroy C. Destroy C.
No, you keep C.
Because it's going to inhibit the killer
The
cells.
virus is getting rid of
Sure,
the
sure.
So HIV has a very clever mechanism for getting rid of A and B, but keeping C. A, B, and C are extremely similar. So to do this is a very clever trick. They're not identical. And that's what HIV takes advantage of. So it gets rid of A and B and keeps C, so it's escaping from the adaptive immune system by getting rid of A and B, and it's escaping from immunosurveillance by natural killer cells by keeping C. Go back. So then some tumors, as I was saying, downregulate histocompatibility proteins in order to escape from the immune system. But they're then susceptible to attack by natural killer cells. And so no one has proven this, but the hypothesis is that they remain dormant as long as natural killer cells can keep them in check. But when they develop another mutation that lets them, whatever that is, that lets them escape from natural killer cells, then metastatic lesions begin to grow out. The topic I'm most interested in research these days is number 2. The role of natural killer cells in the uterine decidua during implantation in pregnancy. So it says there that 10 to 20% of peripheral blood lymphocytes are natural killer cells. They're pretty numerous, but they were really ignored for 20 or 30 years while everyone was preoccupied with the adaptive immune system. So the situation in the uterine decidua is reversed. 70% of decidual lymphocytes are natural killer cells, and only 10 or 20% are T cells and B cells. So at the end of each cycle, a new epithelium is laid down in the uterus, and it's devoid of lymphocytes. But at the midpoint of the cycle, when progesterone begins to be secreted, lymphocytes rapidly accumulate. And they're very numerous, and they represent 70% of the total lymphocytes in the uterus. And then at the end of the cycle, they're shed, of course. But if the woman becomes pregnant, then they are retained throughout pregnancy in the decidua. And they certainly have important roles in what are the various phenomena that have to happen in order for a woman to hold a pregnancy. What has to happen? What has to happen?
The immune system has to be suppressed. The immune system has to be suppressed.
The immune system has to be suppressed. Why is that?
So the fetus isn't rejected.
So what?
So the fetus isn't rejected.
Why would the fetus be rejected?
It's foreign.
What? It's
Foreign
foreign,
body.
right. That's the great enigma of immunology. That the fetus is foreign. It has paternal antigens, but it's not rejected. So if you take fetal skin, a lot of you can do it in the mouse, but in the recent past it's been done in man too, because they do a lot of intrauterine surgery and you can get fetal skin. So if you take fetal skin and make a transplant on the female, she rejects the skin. But she doesn't reject the fetus in utero. So natural killer cells may have a role in that, in the uterine decidua, because they secrete some very unusual substances which may be immunosuppressive. I said maybe, because that's the topic that we're presently working on, and I think other people must be working on it too. There are 3 or 4 unusual proteins made by decidual lymphocytes. So that's one thing. There have to be mechanisms to prevent rejection. What else?
You have to prevent the uterine lining from being removed, the epithelial cells.
Yeah. What else about the lining, though?
It has to be.
It's on the slide.
Has to be permeable.
It's on the slide.
The blood vessels have to reach the fetus. You have to go through the uterine lining.
What did you say? That's
The blood vessels have to get through the uterine lining to the embryo.
It has to do with blood vessels, but that's not quite it. The blood vessels of the uterus have to supply blood to the placental vascular space, where blood exchange, where a nutrient and gas exchange occurs. And so the uterine arteries are spiral normally, so so they're tightly coiled, and therefore the blood flow through them in a normal female is very limited. But when the woman becomes pregnant, the spiral becomes a big tube, and the blood flow increases manyfold. And that's one of the major events that happens in pregnancy. And it's quite an amazing phenomenon. There's no way to study it, unfortunately, that I can think of, because placentation is still undergoing evolution. And the form of placentation in man is only shared by the great apes. Women are difficult, but the great apes are much more difficult. So we're restricted to studying Homo sapiens, and we can't do very much. It's a problem. Preeclampsia, you know what that is? What's preeclampsia?
So when the part of the wall lining starts to separate?
No.
I think it's high blood pressure.
Somebody said it.
Constriction.
Yeah, it's high blood pressure.
High blood pressure, right. Is anyone in the room who has children have preeclampsia? So it's life-threatening. It occurs only with the first pregnancy. It has to do with the failure of the system to dilate the spiral arteries. But there's another enigma. If the woman becomes pregnant with another partner, she can get preeclampsia again in the first pregnancy with the new partner. Yeah, it's too hard to study. That's the problem. It's really interesting, but there aren't any experimental animals that you can use to study it. Natural killer cells probably have something to do with it, with the whole phenomenon. The natural killer cells, the third item on the bottom of the slide, is that they produce cytokines. I'm sure you know what cytokines are. And we probably don't know all the cytokines that they produce. Do you know what microarray analysis is? I'll educate you in a couple seconds. Oh no, it's on the disk. There's not a projectors there, but
I can get a computer if you want one.
that's okay. I can just talk. So, microarray analysis is a technique where you copy all of the RNAs made by cells. And you then put on a chip oligonucleotides that correspond to all the genes that you know about, all that you know about. It's really important to remember that there are lots you don't know about, so they're not on the chip. You take the RNA and you hybridize it. This chip's a little tiny thing, and you can get 30,000 genes on it. It's really quite amazing. 19 different probes from 30,000 genes can go on this little microchip. And then you put the RNA on, and you can determine which genes are being expressed. RNA is the messenger, so the RNA is representative of an expressed gene. So it turns out that natural killer cells in peripheral blood and those in Decidua differ by about 3,000 genes. That's really different. By expression of about 3,000 genes. So the natural killer cells in the uterus are very different from the ones in peripheral blood. They share some features that result in their both being classified as natural killer cells. But the ones in the uterus are substantially different. They're in the same lineage, but they branched apart early from that lineage.
Are they produced in the uterus or are they modified in the uterus?
That's a really interesting question. We don't know where they're produced.
Is it just during pregnancy also?
What?
Is it just during the pregnancy
That what?
that they're different?
No. Oh, probably not. There are, how do I say this? There aren't many women willing to donate uteruses to study it. So great apes are even less willing. Animal conservationists don't let you near them. So, but we're actually involved in collaboration with a group at Dartmouth Medical School that's trying to get us material from normal women. So if you have a big enough OB/GYN service, then there are enough hysterectomies at all ages to get material. So that's our source, but we're not sure we can get enough from a single uterus, normal uterus. It's also, if you're getting random uteruses at hysterectomy, then they're all different in what part of the cycle they come from. So that's another problem in studying it. So we're not sure it's a feasible project, but we're making an attempt. There's one other phenomenon that it is on the slide.
It's just missing. I can see the
What?
top of the form.
Somebody ask something? The other interesting phenomenon is implantation. What happens on implantation? And what are the factors that allow a blastocyst that has partially developed in the fallopian tubes to implant? So that's probably failure of implantation is probably a prominent cause of infertility. We have the daughter of a close friend of ours can't get pregnant, and she has multiple chemical pregnancies, no miscarriages. Chemical pregnancy is a pregnancy where they can detect a urinary gonadotropin, but there's never any miscarriage, and there's never any maintenance of the pregnancy. So you know she gets pregnant because gonadotropin starts being produced, but then no implantation occurs. So that's probably a fairly common cause, and we think that the decidua has to be prepared for implantation in some way, and that natural killer cells may have a prominent role in secreting cytokines that may be involved in implantation.
Dr. Strominger, is ectopic pregnancy more common in some families than others, and would that be related to this at all?
I don't have any clues.
Okay, I mean, I was just curious why why the blastocyst could implant in the fallopian tube if that had anything to do with these natural killer cells or anything like that.
Or why did it plant in the fallopian tube or in the peritoneum?
Right.
Yeah, that's a good question. Something, though, is needed for implantation. Maybe it's provided by the blastocyst. Maybe the blastocyst provides something that allows differentiation of the decidua. Okay, let's go to the next slide. So here's how the system works. This is immunosurveillance for a cell that has lost histocompatibility proteins. Tumor cell, for example. Maybe this is a tumor cell or a virus-infected cell that doesn't have any histocompatibility proteins on the surface. So, and this is an inhibitory receptor on the natural killer cell. So if the ligand for this receptor is missing, then this cell gets killed. Or in the allogeneic situation, if the wrong ligand is present that doesn't fit this receptor, the cell will again get lysed. But if the cell has got a histocompatibility protein that fits that inhibitory receptor, then some sort of signal gets delivered to the lysis system that prevents lysis. So inhibition is dominant. So even if you have an intact lysis system, if you have an intact inhibitory system, no lysis occurs. This little yellow ball is the ligand on the target cell for the lysis receptor. And that's another enigma. People have been trying to figure out what ligands are for 10 years now for natural killer cells. Nobody knows. They have to be very highly conserved substances. So that's how natural killer cells work. If you don't remember anything else, remember that there are natural killer cells in the uterus that are different, and that this system operates by a 2-ligand, 2-receptor system. One pair is activating and leads to lysis, and the other pair is inhibiting. Let me see what else is here. There's a lot more slides, but I don't want to get lost in detail. I can show you how it was discovered. This system was discovered in Italy. When people were studying large Italian families. Here's a large Italian family. And black means that that cell can be lysed by natural killer cells. And white means that it can't be lysed. And if you look, you will see that wherever there's an A, So A and B are haplotypes. Do you know what haplotypes are? Haplotypes refer to, they really refer to the MHC type, to the histocompatibility type. So each chromosome has a different histocompatibility type. They're called A and B, and C and D. And if you look, you'll see whenever there's an A, there's no lysis. A carries the dominant inhibitory gene. I don't know how much you know. You want me to try to explain this to you? So this shows how different natural killer cell lysis is from T cell lysis. So let's consider this child and his or her parents. And you can see that the cells of the child are lysed by that panel of natural killer cell clones. But the parents' cells are not lysed. And so that says that both of the parents have one inhibitory haplotype and one susceptibility haplotype. And so they're both resistant. But this child inherited the two susceptibility haplotypes. So inhibition is dominant. It's quite different in T cells. So here are T cells. You can see that this parent is not susceptible. This parent is susceptible to that clone. And now sensitivity is down. So if you inherit the MHC genes, the histocompatibility genes from the parent, then your T cells recognize those histocompatibility genes and carrying out the lysis. And the same is true for this other. And this was the key discovery. It was Romano Ciccone was the name of the Italian scientist, a young guy who came from a village in the mountains somewhere. And he started to clone natural killer cells before they were really known that they were natural killer cells or what they were doing. And he got this panel of clones, and group 1 and group 2 were dominant. That is, most of the clones he got belonged to group 1 and group 2. And what he saw when he took a panel of targets was that the clones he had obtained could be divided into two groups. Group 1 would kill some targets, and group 2 would kill some other targets. But there were no examples of clones that would kill both targets. Both would kill the targets that were killed by group 1 were never killed by group 2. There were some clones that were not killed by either group of targets.
Can we wrap up with some questions in a few
What?
minutes? Can we wrap up with some questions in a few minutes?
Yeah.
If you have.
Okay, let me see what else is interesting. Okay, I told you that there were 3 class 1 loci called A, B, and C. And it was mapped to C. The inhibitory locus was HLA-C. And somebody in my lab, turns out to be one of the best young people that's passed through, looked at, these are the sequences of the various alleles of HLA-C, or partial sequences, between residues 74 and 89. Who sees anything there? I didn't see it, but he saw it.
The difference at 80?
It's highlighted, so it's easy to see. It isn't highlighted. It is.
There's a difference at 80.
What?
80 is
80 is bold type.
it's K's at the top, and the inhibitors and N's and
So
the
77.
K1
At the
inhibitors.
bottom. So these HLA-C is dimorphic. That means that there are 2 varieties of HLA-C. They either have N and K or S and N at 77 and 80. So that's too complicated. So what it turned out is that there are 2 types of inhibitory receptors. One for one type of HLA-C and one for the other type of HLA-C. So if you have in your cells 2 of one type, then one group of clones will lyse you because you don't have an inhibitory receptor that fits that type. And if you have 2 of the other type, another group of clones will lyse you because you don't have an inhibitory receptor that fits that group. It's a very important system. I'm reviewing a paper it's in my briefcase that says that this system is critically involved in preeclampsia. Something about this inhibitory system. Why is that a possibility? The preeclampsia could be related to this system.
Because now you have 2 parental factors.
Right, but it's because the fetus is carrying one C gene from the mother and one C gene from the father. So the mother's cells weren't generated to respond to that system. If you have the wrong match, if the husband has the wrong C allele, then you can get into a situation where there's a mismatch at the maternal fetal interface in the system. And I can't understand how it could be involved in preeclampsia. And I don't think the people who wrote this paper understand that either.
How would you explain success in implantation of a foreign fertilized egg fetus?
A foreign fertilized egg, that's, you mean in a surrogate mother?
In a surrogate mother, yeah.
Right, how do you explain that? Well, the system already can adapt to one foreign set of proteins and can also adapt to 2 foreign sets.
So it's
The rate of preeclampsia goes way up
Oh, that was the question.
in surrogate mothers. Yeah. I think I saw the number and I can't believe it. The number I saw was that 30% of surrogate pregnancies end up in preeclampsia. That's the number I saw. Okay, questions?
Is placenta previa, is that any relation to natural killer cells? Or,
Placenta previa, is that where
I
the
think it's,
placenta is in the wrong place?
yeah, if that's just a failure in how it grows, but it has
I
nothing to do
don't know.
with
It's a
blood.
failure in it's a failure in selecting the place for implantation.
Okay.
It's selecting the wrong place for implantation. I don't have any clues.
This NK cell started with work on transplantation rejection? Or did I mix
You
that up?
you mixed it up.
I'm sorry. That was,
The work on the histocompatibility protein started with transplantation. And how did the work on natural killer cells really started with finding cells and try to keep them in the right
Oh,
order.
yeah. Done this before.
Really started with tumor surveillance. Actually, in fact, Eva and George Klein, Eva Klein discovered natural killer cells and named them. They were Jewish, Hungarian young people, and they had to flee. They fled to Sweden. They've done all their work at the Karolinska Institute, and their 80th birthday is this summer, next summer, next June, and I'm invited to a meeting to celebrate their 80th birthday. They're wonderful people.
well, thank you, Dr. Strominger.